Hello and welcome to this video on chimeric research. This is not the chimeric research of Fullmetal Alchemist, but instead it more accurately references the mythological origins of the term. A chimera is made up of cells from multiple species. It could be a mouse with human cells, or a monkey with human cells, pigs with monkey cells. These all exist alongside the normal cells of the host organism. Most chimeric research is used to explore embryonic development, creation of functional organs that are from another species, or for stem cell research. In Greek mythology, the chimera had the head of a lion, the body of a goat, and the tail of a snake. In research, it's somewhat different. The embryo is allowed to develop for several days to a week. This leads to hundreds of cells that are the original species. At this point, a small number, and often less than 50 cells from a second species are added. These new cells are pluripotent stem cells, can turn into anything. That allows them to be used the same as most of the first species cells are at this point. The last part is important. Historically, incorporation of the second species of cells was poor. It is only now that we have much better grasp of the process that integration is even remotely acceptable. The earliest chimeras were developed in the 1960s. Recent research has demonstrated how this is difficult, no matter what you're doing. At day 6, 132 embryos were alive, and this is where they added the human cells. At day 10, 103 had survived, and at day 19, only 3 had survived. Although, as these were chimpanzee embryos, they can only survive for 20 days outside the uterus, so there is a hard cap on how long this experiment could run for. This sort of creation is important to understand several events. There is the aforementioned developmental and differentiation process. There is also the communication and integration process between two cells of different species. This part is important, as the two different species interactions are like two people trying to work together but only able to speak in different languages. The further apart the species are, the further apart you could compare languages. The team that was doing this was trying to uncover what happens when you have signals between these different cells from different species, and what is responsible for these different signaling actions. Can they be somehow reconciled, and can these cells then work together? Chimeric research is important for roles beyond what we have spoken about. The most fundamental of these is how chimeric research is far better as a way to test drugs than something like humanized mice. Humanized mice have human genes that have been transferred. These still work within the mouse cell, and are only a small number of genes. The chimera has actual human cells. These have a full human genome. They should behave and respond in the same way that cells would in a human. This means the results you get from a chimera are going to more accurately recreate what you would see in a human than what you see from the humanized mouse. This point is important when you consider how many drugs, or at least drug leads, do not make the jump between animal model and clinical trials successfully. This is down to a range of factors, but part of it is the differences between species. The most recent research that we are aware of found the following results. There is an average of up to 4% of the cells in the embryo that were transferred being human. This means it's 96% chimpanzee and 4% human. Most of these cells, about 5%, were in the innermost layer. That means most of these cells would have found their way into the fully developed organism if it had gotten to that point. There was almost none of the human cells found in the extra embryonic tissue, that is, things that would form the placenta. If you were to look at this as trying to develop an organism for research purposes or for medical treatments, all of the cells that have been put into it would become part of the living developed creature. This carries with it a whole raft of ethical issues we won't get into now, but understand that chimeric research does come with problems, and these problems are much like what you see with normal animal research, but they're even more difficult to untangle, primarily because you're looking at not only an animal research project that may have things like consciousness, it may have reproducibility, and more. Chimeric research is limited at the moment by being in its infancy. Despite having more than 60 years of research into it so far, we still struggle to create effective and efficient chimera models. This can be seen with the attrition in chimeras, which escalated quickly over a mere 13 days. In 13 days, you went from 132 embryos to 3. At the same time, this research poses pathways to develop things like synthetic or artificially generated organs, ways to minimize the number of animals needed in research, ways to ensure that drugs as they develop will have accurate representations in the human response. There are a lot of benefits to the chimera, and ultimately there are a lot more questions it's going to raise as well. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.